Okay, so we are now recording. Welcome everybody to the Fungisphere group meeting. Today we're very lucky. We're going to have an update from David Phalen on some of his research interests, and this will be available for people to view online later on. So David, I'll let you do your own introductions, and we look forward to hearing what you have to share. So thank you.
All right, my name is David Phalen, and I've been an avian veterinarian and an infectious disease investigator, largely around pet and exotic birds, but also wildlife since the late 1980s. I spent 17 years at Texas A&M in the faculty there after finishing my PhD, which was on viruses, and then joined University of Sydney as the director of the Avian, Reptile, and Exotic Pet Hospital in 2006. And I started this work in the late 1990s, and there's sort of the Texas A&M phase of it all, and then there's continued work that we've done here in Australia. So without further ado, I'll get on to the topic today, which is Macrorhabdus ornithogaster. So most practitioners would first encounter Macrorhabdus in a wet prep under the microscope, either of feces or at a bird that had died and they had done a scraping of the isthmus, which is the junction between the glandular stomach or the proventriculus and the grinding stomach, the ventriculus. And you can see that these organisms are long rod-shaped structures that are very, very stiff, have rounded edges, and are a bit refractile under the microscope. The Dutch were the first to describe these, and they called them in Dutch, which I don't know what the words are, yeast sticks. I think the Italians call them some sort of bread, but anyway, we find this is an organism that's been found all over the world. It's got a completely global distribution. Anywhere where there are aviary birds, this organism is found. And it was originally found in finches, canaries, and in the Netherlands, followed by native finches that were kept as pets, and then found in wild finches in Europe and the UK. But the species that we see it the most in practice are Budgerigars, Lovebirds, and finches. It's been described in Israel in ostrich farms, and it's been sporadically described in chickens in North America, Europe, and we've seen it here in backyard chickens in Australia as well. It occurs in wild birds in Australia, and so we do see wild birds coming in with this infection. There are two species on the left are the European goldfinch and the greenfinch, which are introduced species into Australia. And in birds that have come through the pet trade, these are captured from the wild and then sold in pet stores. We've had virtually all of them have been infected. Now, whether they got infected in the wild or whether they were infected during the process of coming into the pet trade, we're not sure. But we see about 3 or 4 species of parrots commonly infected with this on the east coast of Australia and on the west coast of Australia, and that includes the Galahs and the ringnecks. This is the typical presentation that a practitioner might see in their clinic. They would be a middle-aged male Budgerigar coming in, appears to want to eat, but is just really grinding the food because his stomach is quite painful. They're vomiting, dehydrated, they have weight loss, and they may or may not have diarrhea. And if you do a wet prep, you'd find that of their feces, there'd be large numbers of these organisms in the droppings. The wild birds that we see that come in with this disease are just typically have what we call wasting disease, juvenile Galahs, juvenile king parrots, juvenile corellas. They have fetid feces, and they have multiple different agents that are present that may be contributing to this. They have, a number of them have a spironucleus parasite of the digestive tract, which may or may not be present in all of them. A lot of them have Macrorhabdus, but they don't all have them. Many of them are immunosuppressed with the cytosine beak and feather disease virus. And they all have villus atrophy of their digestive tract, which probably contributes to their diarrhea and a maldigestion, malabsorption syndrome. And obviously the Macrorhabdus doesn't live in the intestinal tract, so some other disease is going on with these birds. And they may have other diseases that are also consistent with immunosuppression. At post-mortem, this would be the proventriculus and ventriculus on the right is a fairly normal one. This is the proventriculus, the top, the skinnier portion, and then the muscular ventriculus down below. And this one on the left has, you can see it's quite distended. There's seeds in the proventriculus, which there never should be. They should all pass directly into the ventriculus. The ventriculus is somewhat distended. If you open it up, you'll find that there's quite a bit of mucus in here. And if you do a scraping right at the junction between the proventriculus and the ventriculus, you'll find massive numbers of organisms. And this is just another bird with a really distended and dilated proventriculus, some thickening and enlargement of the isthmus between the proventriculus and the ventriculus. Oftentimes, these birds will have disruption of the coilin, which is the grinding layer of the inside of the ventriculus, and they'll have punctate ulcers either in the proventriculus or in the ventriculus or in both instances. And when there's gross disease, such as we're seeing here, there'll be millions and millions of organisms on a scraping of the mucosa. Microscopically, they penetrate, the organisms form these sort of log jams that you can see. It's a PAS stained section through the isthmus of Budgerigar. And you can see these organisms sort of stacked up in a log jam. The presence of organisms doesn't necessarily prove disease, but generally when there's lots and lots of them, they penetrate into, they go beyond the isthmus down into the ventriculus, where they penetrate into the ventriculus, cause disruption of the coilin, elicit an inflammatory response. And in most instances where we see those kinds of changes, those birds are not feeling well and have a history of not doing well or thin. And we think that they have a primary disease as the result of this infection. An interesting phenomena is that recently there's been a paper published that has associated this infection with malignant transformation and the formation of gastric adenocarcinomas. And we have seen that in the United States in one flock where we had many dozens of birds brought into us with all ranges of hyperplasia, dysplasia, and then transition into adenocarcinomas. And how these organisms do that, we have absolutely no idea. But occasionally that occurs, and in some instances it can be quite common in certain flocks. So what is this long rod-shaped organism? Well, a good friend of mine was Jerry Dornstein in the Netherlands, and he was working on this for his PhD. And he was about to put together a paper where he was going to describe this as a yeast. He already described it as a yeast in some of his talks, and Linda Lowenstein in California had already described it as a yeast. But Van Herkt was his supervisor who told him it was a bacteria, and to put his notes in his desk drawer and to forget about it. And so Van Herkt described this as a megabacteria, even though it was PAS positive. And he said that on EM they couldn't identify organelles, which in fact they had already. And they published a paper on that. And that name, megabacteria, stuck with this organism till today, actually. There was some additional work done by Scanlon and Graham was my supervisor for my virus stuff. But this work was done while I was there. They cultured a bacteria out of the proventriculus of some budgerigars, And they decided that it was in fact the same organism that they were seeing grossly. But it changed in morphology. They never did a PAS stain on this. And later we went back after he had retired and tested all of his cultures, and they all were true bacteria, and they weren't the yeast that we've subsequently found that they were. So this was all really bad science. There were several attempts to grow this organism during the 1990s, and no one was successful. They talked about it growing and doubling in size over the course of 2 weeks, but I think what they actually saw in some of those cases was the organism would actually fall apart, and they would form 2 organisms instead of 1. So when they did their counts, as you'll see, these rods are actually multiple cells. But they weren't actually dividing, they were just separating into individual cells. So it appeared as if there was more organisms when they got done than when they started, but there wasn't. So what do we know about this organism as far as the way it stains? Like a fungus, it stains positive with PAS. And we were the first to use the chalcofluor stain, which binds to chitin. And chitin is only produced by eukaryotes. So we knew immediately that this was not a bacterium. The gram stain, they call it gram positive, but you can see it only stains the cytoplasm. The gram stain only stains the cytoplasm and not the cell wall. And if you put a Giemsa stain on them, then you can actually see that they have nuclei. And so this is one rod, but there's actually 2 different organisms in here, and you can see the nuclei in there. So we knew it wasn't a bacteria. So we couldn't grow it ourselves, or really didn't spend very much time trying. So we developed a rather crude purification method to separate the organism from any extraneous DNA that might be present in it in the scrapings. We extracted the DNA and sequenced the rDNA, and then did molecular phylogeny on that. And to prove that the sequence that we got out of this organism was in fact coming from that organism, not anywhere else, we made probes using that sequence, and used these probes, which I've never used since, but it was the only thing that we could find that would actually penetrate the capsule wall. It's this peptide nucleic acid probe, which is a combination of a nucleic acid probe attached to a hydrophobic protein. And we were able to show that molecularly, that it was an anamorphic yeast, an anamorphic Ascomyces yeast, which you can see right down in here relative to some others. And that when we stained it with our RNA probe, it lit up just like you wouldn't believe. And you can actually even see the nuclei where there wouldn't be expected to be any RNA. And this was our unstained on the control on the left. And then once it was stained, you could see that only the Macrorhabdus showed up, and that the contaminant yeast, which we have one right here, did not stain. So it's very specific for our organism. And we named it Macrorhabdus ornithogaster, which in Greek means big rod from a bird's stomach. And it's a new genus and a new species, and so far it's the only member of its genus identified to date. So the next question became, why was it so hard to grow? And we had a absolutely lovely person from Japan come over. Hannah Fusa was bound and determined that she was going to grow this. She had 40 experiments set up to go the first week that she came into the lab. And we had it growing in a week and a half after she started. She didn't even stop for jet lag. And one night I came in to pick her up at the lab, and her head was flat down on top of the lab bench and completely asleep. But we used a number of different combinations of broth, solid, agar, various combinations of pH, aerobic and microaerophilic conditions. And what the bottom line is that this organism only grows in microaerophilic environment. It only grows in tissue culture media that contains fetal bovine serum and sugar. and it only grows between a pH of 3 and 4. And it grows best at the bird's body temperature, which is 40, but it'll still grow at 38. So this just shows you the various pHs and how well it grows. And the one that was kind of interesting to me is this pH 3 or pH 2, where we didn't think that it could grow at pH 2. And what we found was that when we started to see it grow, we retested the pH of the media, and it was no longer 2, it was up to 3.5. And so this organism can actually modify its environment to create a pH that's ideal for it to grow. You can see with this that it prefers the 42 degrees centigrade temperature to grow in, and grows at animal body temperature, but at a much lower rate. And that it will grow with 1% or glucose or sucrose, but that it prefers the higher concentrations of the glucose and sucrose. And that when you give it the fetal bovine serum, it really enjoys large concentrations, up to 20% of the growth media being fetal bovine serum. So one of the things that we also found, which we were feeling pretty good, proud of ourselves, we were growing buckets of this stuff. No one had ever been able to grow it before. And then all of a sudden, at about 20 passages, it stopped growing. And we've confirmed that over and over again. And so in the United States, our isolates will be able to be passaged about 20 times. And then there's some essential nutrient that they no longer have. And in Australia, well, I can only get them to passage about 5 or 6 times before they stop growing. So there's something missing in our media that they bring with them, but after multiple passages, they run out of it. And we haven't figured out what that is yet, but we haven't spent an awful lot of time trying. So then we did a chicken infection trial to fulfill Koch's postulates. We isolated the organism from Budgerigars, infected chickens with a pure culture. And with day 1 old chickens given 100,000 organisms, we can get 100% infection of those chicks. We've re-isolated the organism, resequenced the DNA, and during that process, we also had chicks that were not affected, co-housed them with the chicks that were infected, and demonstrated horizontal, probably fecal-oral transmission. Now that we could grow it in culture, we did some in vitro antimicrobial sensitivities. And what we would do is put a certain number of organisms each into one of these 96 well plates or fewer well plates. And then we would mix them with different concentrations of the medicine that we wanted to test them against. And then look, and we would just read these out after 5 days using the spectrophotometer. And if nothing grew, then they would come out with low spectrophotometer readings. If something grew, then they would come out with higher ones. And we looked at fluconazole, which you can see is only really effective at fairly high concentrations. And we've actually treated Budgerigars with fluconazole, and we end up making them sick before we can get a good effect on the Macrorhabdus. We've shown that they are susceptible to amphotericin, at nystatin, but at relatively low, at relatively high concentrations. And as was reported prior to our work by Louis Filipich from University of Queensland, was that that amphotericin, they're quite sensitive to amphotericin B. We tried some low toxic antifungal chemicals, potassium benzoate, potassium sorbate, sodium benzoate, and found that they were all very quite effective at killing the organism in vitro. And we've done some trials with those in vivo. And the problem with that is it does kill the organism, but you have to give the birds the medication continuously, and they end up with either potassium or sodium toxicity. So doesn't work out very well. So pretty much everybody around the world uses amphotericin, either a product that they get compounded at the chemist or a product that that's made commercially for putting in the drinking water here in Australia, or they take these fungal lozenges and they dissolve those and give them to the birds. And our impression was that things weren't working out all that well for our birds. And so we did a retrospective study of the birds that had come in that had been on amphotericin treatment. And we found that only 17% were successfully treated of the 36 cases that we had treated. So another work that was done in Germany, I think they found, I don't remember, it was 40 or 60% success, but it's clearly not working as effectively as we thought it was going to. And so we actually treated two birds, a lovebird and a Budgerigar, that we knew that were shedding Macrorhabdus, that had been shedding for a long period of time, with oral amphotericin compounded by a chemist, given twice a day by gavage for 30 days. And you can see in both instances that you see a dramatic decrease in shedding, in the droppings, as long as they're on the medication. But as soon as you stop the medication, or within 2 weeks after stopping the medication, then the shedding starts again, and even gets even more dramatic than what it was prior to the initial treatment. So again, these are two birds that had temporary cessation of shedding, appeared a little bit better when they were under the treatment, but the infection rebounded right away. So another example of treatment failure. And one of the reasons, one of the important things of this also was showing practitioners that you had to wait at least for 2 weeks after you stopped treatment to know whether it was successful or not, because if you went back the next week and tested, you might not find it. But if you waited for a month, then you'd find that it was back again. So some of these people might have thought that it was successful, but if they'd waited and retested a little bit later, they would have found that it wasn't. So we then did an experimental trial where we infected chickens, day-old chickens, and gave them 2 doses of amphotericin at 25 mg per kg and 100 mg per kg. And we showed that both resulted in reduced shedding, but there was no cures after 10 days of treatment in the chickens that got the 25 mg per kg. And we only got a 30% cure with 100 mg per kg at 10 days, which is what most people are treating with. It's possible if we'd gone out to 100 days, we might have been more successful at cure, but we're not 100 days, 30 days, a month. But at the treatment protocol that most people were using, we're getting at least 70% that remained infected after 10 days. So the other thing that we did then after that was to look at the most common treatment that's used in Australia and around the world. And that's this megabacteria stuff, which is an amphotericin product that's said to be water soluble, and that the Budgerigar breeders and veterinarians around the world can get this. And we treated them according to the manufacturer's recommendation. We had 16 birds that had been shedding for an extended period of time, so we knew that they were clearly infected. We treated them for 10 days and determined if they were still shedding. If they weren't, then we put them to the side and kept them separate. And then we tested them 10 days later. If they were still shedding, then we gave them another 10 days of the amphotericin in the drinking water. And so the birds, out of the initial 16, we had 11 that were negative after 10 days, but of those, 4 became positive again 10 days later. And the ones that we treated for 20 days, 2 out of 5 were negative after 20 days, but 2 were still positive and 1 died. So we had only an overall treatment success of 56% in these birds. And this is what we're seeing consistently with treatment. We're having some birds that respond really well to treatment with the amphotericin, some that it sort of beats it into submission, and then it pops back up. And then the others, it doesn't seem to have very much of an effect on at all. Okay, so clearly we need something better than what we've got to treat these birds. And but what we also know is that a lot of these birds have other factors that are going on in them, which may really complicate the treatments. So many of these birds probably have beak and feather disease infections. We know that they have polyomavirus infections. We know that a fair number of these have trichomonas infections. We know a fair number of these have coccidia infections. And whether any of those are playing into this or just are another sign of a really immunosuppressed birds, and that they're not going to respond just by treatment alone, that there needs to be a good host immune response in order to get rid of this organism, we don't know. But we suspect that there's a lot more to this. It's a lot more complicated than just a Macrorhabdus or not. So we've done some genetics work on these, and we're in the process of trying to get enough data together to look at the epizootiology of this and its spread across the world. What we know is in Australia that most of the isolates are very, very similar in the approximately 1,500 nucleotides that we sequence in the ribosomal RNA. We're only finding 4 point mutations that are typically found in these birds, sometimes up to 6. But we found something that's very interesting that I've not seen in a yeast before, and not been able to find in a yeast before, is that this organism has the ability to acquire its host ribosomal DNA. And so one of the first ones that we sequenced that was a little bit weird was this one from a canary, and we had this section here in the 18S that when we blast it, matches up with DNA from multiple different other bird species, some from Europe. We think that this bird has probably acquired host DNA from one of the birds that it's infected sometime during the history of its ancestry. But then we found the second one from a zebra finch that not only had this first insert, which belongs to one species of bird, but had this second insert in its rDNA that comes from a second species of bird. So he's been in this bird, whatever it is initially, and then subsequently infected another bird and picked it up, another bit of the host rDNA there. And our next steps are to do whole genome sequencing and to look to see if there's any other evidence of their abilities to pick up host DNA. And then we're also trying to get more data from organisms around the world again, so that we can look at how this might have moved around and where it might have started. Did it start in Australia or did it start somewhere else? Chickens grow pretty fast and get pretty big and aren't really the ideal treatment model because the amount of drugs that we have to put into them as they're growing gets quite high and very expensive. So we're probably going to look to using a new model, a quail, which will only get to be about 100 grams, and see if we can do infection trials in those and then use those for our treatment trials. There's been some work that's shown that Amphotericin and combined with other drugs and some simple things like vitamin C might be more effective. And we have done a couple of birds, treated a couple of birds in the clinic with that, and the Macrorhabdus has gone away. Whether that means anything or not, I'm not sure at this point in time. We're looking at delivery forms and methods that might result in a longer persistence of the medication at the junction of the proventriculus and the ventriculus where the organisms live. And if we can figure out what we need to keep them going in culture in Australia, we can do a lot more in vitro testing and see that way perhaps to identify some better treatment options than what we have at this point in time before we go to the in vivo model. So there was a lot of really talented and bright people that were involved in this, and Elizabeth Tomaszewski was the first one to suggest PNA probes. Hannah was the one that just worked herself to death to find a way to grow this for the first time, and Allison did all the work that was related to the in vitro testing, antibiotic testing. Hamish is my current student who's been doing the work that we've been doing here in Australia, and Kitty was an honors student that helped him. We've received a lot of support in funding from the Schubot Exotic Bird Health Center, Texas A&M, and the Sydney School of Veterinary Science. So I haven't been looking at any comments. I'll look at those now, and I'm happy to take any questions.
Thank you, David. That was a really interesting talk. I've got several questions. I haven't seen the chat box just yet, but first of all, I'll give anybody else an opportunity to fire with some initial questions. No, we have an apology, that's all, in the chat box.
Yeah, sorry, Justin, I was just going to ask, thanks, David, that was a really nice talk. I guess I keep drawing parallels between your organism and Cryptococcus neoformans, the Basidiomycete. So, I mean, obviously that one doesn't, well, I don't think it survives very well in the stomach, but it's found, I mean, there's a theory that it goes through the bird's digestive tract, and it's found quite copiously in guano because it's got those nitrogenous compounds that it can use. But it's just interesting how, like, crypto doesn't sort of adopt that niche in the stomach that your yeast does. So it's almost like your yeast has adapted to low pH, perhaps much better than other yeasts. And I just wondered if you had any comments about that.
Well, I think mostly crypto has been associated with the droppings because especially in the United States, but in other parts you'll have these, 10 million starlings and various other species of blackbirds that'll get together and roost in the same place. So they produce large amounts of feces, and it's a great place for the organism to grow. We do see crypto in birds in Australia, and it causes the same kind of systemic disease that you would expect to see in other animals. It localizes in the upper respiratory tract, in the beak, causes marked deformities of the bill. So I don't know how much correlation we can put between the two and why crypto doesn't like the stomach of birds, but we don't see it developing in the digestive tract.
Okay, so you see respiratory disease with yours as well?
No, we don't.
No, okay.
Only Cryptococcus.
Yeah, right, okay.
Disease in birds, but ours doesn't. So there was a couple of notes in the vet record that said that they saw an organism like this in the nasal discharge from the of a cat and a dog. They didn't show any pictures. They didn't do any cytology. They didn't do anything. We had to believe them that was the case. And there's a lot of speculation that this organism can grow in mammals. So we've done that research. I didn't include it in this talk, but Hannah came from Japan down here, and that's one of the things that she wanted to do straight away. And we inoculated mice through the respiratory tract and through the oral cavity and intraperitoneally, and we were unable to establish an infection in mice.
Okay.
Interesting.
Thank you.
Richard has asked a question about the use of triazoles and natamycin for therapy. I know you mentioned fluconazole, but have you tried any of the other azole agents?
No, we haven't.
Do you think there's any point?
It's worth trying a lot of different things, I think, at this point in time. I think definitely, because what we have now is not working.
Yeah. And so I had a question about whether on autopsy of your birds, is there any tissue invasion? Is there any other sort of sites where the infection could reside that's not being treated by the purely interluminal amphotericin?
Yeah, well, the mucosal glands of the proventriculus, or the isthmus, the junction between the proventriculus and the ventriculus, are very, very tight. Each little villus is right up against the other, and they penetrate down deeply into the crypts of those glands. They don't penetrate into the tissue. When there's a severe disease, it penetrates down into the colon, and the colon is a dense carbohydrate layer that's produced by the glands in the ventriculus. And I'm quite confident that down deep into that colon, they would be well protected from any medication that's given to them orally. And that would be what you'd have to do, would be to continually treat them as that colon grows out and sloughs off to get rid of them. So that's why probably much longer treatment protocols are more likely to be successful.
Mm.
But we treated for 30 days in those birds, and the organism came right back. So it's hiding somewhere. It's avoiding the drugs. There's no question about it.
Yeah, and obviously we see, crypto, sorry to keep comparing, but crypto requires really long treatment courses, right? So induction is 2 weeks, consolidation is 8 weeks, and maintenance is normally up to 6 months in non-immunocompromised hosts. So eradicating that yeast infection takes a really long time.
I don't think anybody's had the patience to treat birds for 6 months, but you never know.
Yeah. I have other questions, but is there anybody else who's kind of waiting to ask or type a question in? Otherwise, I'll carry on. I'll carry on. Please unmute yourself if you have a question. So you kind of touched on a little bit about these birds have other infections generally, and maybe sick immunocompromised birds. So to what degree do you think this is a pathogen, and what degree is a marker of ill health?
Okay, well, when we've done our chicken infection trials, we've monitored weight gain over the course of the time, and we've clearly shown that there's about a 10 to 20% reduction in weight gain in birds that are treated, that have been inoculated with Macrorhabdus, compared to controls that have no infection. And these birds are healthy white leghorns that would not have any other, they're specific pathogen-free chickens. So we know that it does have a primary, can have a primary effect. We certainly know that a lot of these birds that are having the signs of regurgitation and everything, once you treat them with for the Macrorhabdus, that the ones that do respond to treatment get better, the organisms go away, and they don't come back if they're isolated and not exposed to it again, and the birds can go on and do well. So I think in some instances, it definitely is a primary pathogen. But I guess one of the things that you have to be aware of, and one of the things that's been very frustrating to me that I've finally learned to live with, or just don't care so much about, is that the bulk of these Budgerigar breeders and these large aviaries where they have all these problems have absolutely no concept of hygiene, have no concept of isolation, have no concept of quarantine. And so they'll have their sick birds in cages on top of their quarantine birds that'll be in the kitchen, that'll be next to their big flights where they're breeding their birds. And they take their birds to shows, and they mix them with other birds and bring them back all the time. So this really, I think if I was going to target my understanding of this organism, it would be mostly to treat the individual pet bird that means something to the owners, that you can treat, and you know that it's that it's going to be it's not going to be exposed to the organism again, as opposed to trying to get these flock treatments where right now most Budgerigar breeders will treat their flocks at least twice a year with tetracycline to get rid of the chlamydia, and they'll treat them for a good 10 days or maybe 30 days in the water, which will not get effective therapeutic levels for most birds, be toxic for some, and may get a few therapeutic for the others. They're routinely putting Nielstat in the drinking water and the food. They're routinely treating with this Megabac S, and then, you know, who knows what else they throw in every once in a while. So these are the absolute worst clients, the absolute worst as far as creating antimicrobial resistance in flocks, and the most likely to listen to anybody and anybody about what might work. And, you know, they've got all these things that they're putting in their food and everything else. So I guess it's of interest scientifically. It's certainly an interest from the point of view of the individual sick bird and the person that's not feeling, whose bird's not feeling well, that you want to make better. But we're never going to get to a point where we can develop a treatment that's going to be effective on a broad scale, flock-wise basis, because of a combination of the other diseases that they have and their total lack of knowledge about hygiene and quarantine and concern for disease transmission between birds.
Yeah, gotcha. That's challenging. Megan is asking about the vitamin C amphotericin combination dose that you found to be effective.
So I should have read up on more of this, but I'll send you, I can email you the paper on that. But basically, apparently, they were doing this in vitro, where they were growing the aspergillus, and they felt that the vitamin C would cause a oxidative stress to the walls of the aspergillus, and that would make it the amphotericin more likely to penetrate into the aspergillus itself, and would create a metabolic, little bit of a metabolic crisis for the amphotericin. And they showed that it increased the efficacy of the amphotericin quite dramatically. And so I thought, well, what the heck? We're giving the amphotericin orally. What's a little extra, you know, what's some vitamin C going to do? And if it's going to create an oxidative effect on the Macrorhabdus, maybe that will help. So again, I'd like to get some in vitro studies where we can grow the Macrorhabdus in our in the lab and add the amphotericin in combination with the vitamin C and see what happened. And then maybe we could go to some experimental trials in quail or something to test that.
Yeah. So Julie's asked a question that I was also wondering about, so that's good. So there's quite a lot of environmental existing yeast that have adapted to survive inside amoebae, and it confers an ability to hide inside macrophage. Have you got any evidence that phagocytosed organisms survive inside macrophages with their low pH?
We have no evidence whatsoever that any of these organisms are phagocytized and that they can move around in the host body or anything. There's been a few reports of them being found in the liver, but I'm totally and absolutely convinced that those are false findings and that they were never adequately documented. So the only place that this organism appears to be able to survive is in the lumen of the stomach.
Okay, great. Well, thank you, David. That was really interesting and lots of food for thought, and it's really interesting to see the process of identifying and properly classifying an organism and then starting to look at treatment options. That's really, really useful for us all, I think. We will post this video with your contact details on the Marie Bashir Institute website, so people, I guess, you're happy to be contacted with other questions.
Sure, absolutely.
So just before we close up, and everyone probably has another meeting at 4 o'clock, I do, I wanted to ask a quick question. There's only 9 of us here, but about the ongoing activities for the node for the rest of this year. We plan to have a series of talks which will be related to different themes, and we'll give more notice of those. And at the end of the year, we're planning to have an event as well. And exactly what that event should be is not yet determined, but we're going to have a meeting with the theme leaders to discuss that. And the main options will appear on your screen now as a poll, whether we look at something along the lines of a grant writing strategy workshop, a symposium to share research interests, a kind of collaboration incubator to try and get people generating new ideas together, or a conference style event where we would invite some international speakers. So could I just get you to click which of those you think would be the most valuable? And I will take that information to the rest of the team leaders when we meet next week. And whilst people, you can feel free to unmute yourself and tell me what your idea is, by the way. You don't necessarily have to answer the poll. That's good. Well, we're pretty evenly split between the symposium and the collaborator incubator.
I would think a symposium would also result in collaboration and incubation, but
Yeah, it's definitely possible to combine some of these things as well. Yeah, okay, good. We normally, last year we had a day event and it worked pretty well, so maybe we can combine some of those features. Okay, does anybody have any kind of final comments before we call it a day?
Just thank you to David for giving a really great talk.
well, it was my pleasure. It was good to share what we're doing with you guys.
Yep, thank you, David. I know that there are other people, you know, on campus working on fungi. So it's been great.
Okay, all right, well, fantastic. Thank you everyone who made it, and thank you, David, very much for the talk. It was super interesting, and we'll get this up on the website so that other people can enjoy it too. So thank you very much, everybody.
All right.
For organising,
Stay
Justin.
safe.
Thank you. Bye.
Thank you.
See you all soon.
Thank you. Yeah, see you in Vietnam.